Hello everyone. In this lecture today, I'm going to talk to you about two different types of antiviral agents, that are directly acting antiviral agents versus host-directed antiviral agents. So the overview of the topics that I'm going to cover is what are the different directly acting antiviral agents and what is the mechanism of their action, what are host-directed antiviral agents and what is their mechanism of action, and what are the risks and benefits associated with these directly acting antiviral agents and host-directed antiviral agents. And finally, I'm going to give you the status of currently what proportion of the directly acting versus host-directed antiviral agents are available. Okay. So this chart here gives the summary of different phases involved in the virus life cycle in a host cell. So we'll be discussing these directly acting antiviral agents and host-directed antiviral agents in relation to these different steps of the virus life cycle. Okay, so now let's get started first with the directly acting antiviral agents. Okay, so what are directly acting antiviral agents? So these directly acting antiviral agents, they target viral proteins or the viral factors. Therefore, they are called directly acting antiviral agents because they directly target the viral proteins. These proteins might be important for different stages of virus life cycle. Okay, so when we use directly acting antiviral agents, they selectively target one virus. They are selectively, these antiviral agents are selective against one virus. So that is the actually limitation of these directly acting antiviral agents, because we cannot use them against another virus. If it's a different virus, then we have to find another directly acting antiviral agents. And another important point is that the directly acting antiviral agents, for example, we targeted a protein. Let's say that protein is called A protein, and this is the protein sequence. And if there is some mutation, if here is mutation, and if here is mutation in this protein of the virus, then what will happen is that our directly acting antiviral agents, it will not be able to produce the desirable antiviral effects because the target site has already been mutated. Okay, so this is the drawback of the directly acting antiviral agents. Okay, so guys, once again I'm going to repeat. So what are directly acting antiviral agents? The directly acting antiviral agents are the drugs that directly target viral proteins or the viral factors which are important for different stages of viral life cycles. Most often these directly acting antiviral agents, they are selective against one virus, and these directly acting, and the major limitation of these directly acting antiviral agents is that if there is some mutation in the target protein of the virus, okay, if there is some mutation, for example, if there is some mutation in the protein A, let's say that this is a spike protein. If there is mutation in this protein, then our directly acting antiviral agents, they may not be able to produce antiviral effects because the target site of that the target site of that agent has been mutated in the virus. Okay, so this is the limitation of directly acting antiviral agents. So to counteract or to counteract that limitation, so there are another kinds of agents which are called host-directed antiviral agents. So what is the difference? The difference is that in host-directed antiviral agents, these agents, they target host proteins. Okay, so these agents, they target host proteins or the host factors which are essential for different stages of virus life cycles. And so therefore these proteins or these factors can be targeted for the development of host-directed antiviral agents. Okay, since viruses from one family, okay, so the major advantage of this class of drug is that since the viruses of one family, they often employ the same host proteins. Therefore, targeting these proteins can produce agents. We will have the agents that have broad spectrum antiviral activity, and offer higher barrier to the development of drug-resistant virus strains. Okay. So what does this mean is, guys, okay, so if we have a virus A, virus B, and virus C, if they are from the same, if these all viruses, if they use same target proteins, let's say that they use same target proteins D, and we have our antiviral agents which are actually targeting this target site, okay, this target protein in the host. Okay, so this target protein in the host, this can be the receptor of for the virus entry. So then what is the benefit is that the antiviral agents that is used against this target protein can be useful in all these three viruses, virus A, virus B, and virus C. So that is the major advantage. So therefore it offers broad spectrum of antiviral activity. Okay, so and another thing is that, guys, another important thing. Okay, so another thing that I want to mention is that if we have a host directed antiviral agents, then these agents actually can be employed to the new virus if that new virus uses the same host protein as the other already available virus. So then we can employ this host directed antiviral agents to counteract or for the treatment of this new viral pathogen. The condition should be that this viral pathogen should use a same host factor. Okay. And yes, because another thing that I want to mention about host-directed antiviral agent is that there are over dozen zoonotic viruses. That means these viruses they transfer from animals to humans that have caused deadly human disease in recent years and will remain potential source of future outbreaks. So therefore, if we have host-directed antiviral agents, then these agents can be used against those viruses. And also, one important recent example is that we recently witnessed epidemics in 2012 MERS and in 2016 Zika prior to current COVID-19 pandemic. So I mean, just imagine that if there is a new virus and this virus also uses the same host protein or the factors as the virus that were already available before, and we can employ actually the host directed antiviral agents against this particular factor to deal with this new virus or the new pandemic. Okay, so this is the major advantage of host directed antiviral agents. Okay, so now moving to the next slide. Yes, we will have already available drug that can be used when we have this host-directed antiviral agents, and they can be beneficial in slowing the viral spread in case of outbreaks. This is important point. And host-directed antibody actually they can be complemented with directly acting antiviral agents and vaccines. Okay, to deal with these new outbreaks. Okay, so guys, so then the two main concepts of host-directed antiviral agents, they are that these host-directed antiviral agents, they interfere with the host factors. They interfere with the host factors that are required for the pathogenesis. That means that they target the host factors which are required for the virus life cycle. Okay, these includes host cell entry, replication, and assembly. Another strategy in host-directed antiviral agents is that we can target the host by boosting the host immunity. Okay, so another strategy is to target the host by boosting the host's innate immunity through the administration of interferon gamma, different interferons, or toll-like receptor agonists. Okay, for example. Okay, so guys, as you can see that we can target host proteins or the host factors which are important for virus entry, or via the translation of the viral genome, or for the virus replication, or the assembly of the virus, and also the release of the virus. So these are the different stages of virus life cycle that can be targeted So the different stages of virus life cycles. So we have to find out which are the important molecules from the host, which are important for these different stages of virus life cycle, and we can target those important molecules which are responsible or which are required for these different stages of virus life cycle. So these kind of agents are called host-directed antiviral agents. Okay, guys? So now, more on host-directed antiviral agents. Like I said, we can target the virus entry. For example, host protease inhibition we can do to restrict virus entry. Is an example, different respiratory viruses such as influenza, parainfluenza, and coronaviruses, they rely on host proteases. Why? Because these proteases, they activate the entry factors. They activate the entry factors or the receptors that facilitate membrane fusion and entry into the host cells. Okay. For example, the transmembrane serine proteases 2, TMPRSS2, it is ubiquitously expressed serine protease that is crucial to the cleavage and activation of both hemagglutinin of influenza viruses and the spike protein of the SARS and SARS-like coronaviruses, SARS-CoV-2 and SARS-like coronaviruses. Another thing that will be that we can target is the depletion of intracellular nucleotide pools. Okay, this is also another host factors, the nucleotide pools that can be targeted, and an enhancement of virus mutagenesis. Because when virus replication happens in the host cells, then they require nucleotides, right? So the they depend on the available nucleotide pools. So we can target these nucleotide pools to compromise virus replication. So here we are targeting host factors, not the virus directly. So inosine 5 monophosphate dehydrogenase catalyzes essential step in the biosynthesis of guanine nucleotides. So basically conversion of these IMP to xanthosine monophosphate XMP. So IMP-DS inhibitors, for example, VX497, they have broad spectrum activity. So we can actually target the nucleotide pools in the host cell. Then by targeting these important molecule IMP-DS from the host. And so this is one example of targeting of the nucleotide pools in the host. So a third example is we can also target proviral kinases and vesicular and secretory pathways. A recent genome-wide screening approaches using small interfering RNAs or CRISPR-Cas assays targeting cellular kinome have highlighted proviral cellular factor. Okay, so this, they can also serve as a target for the host-directed antiviral agents. So then we can target these proviral kinases and vesicular and secretory pathways, to have the antiviral drugs. And we can also target sigma receptors and virus-induced ER stress. Gordon et al., they express actually 26 tagged SARS-CoV-2 protein in human cells to identify potential proteins that physically associated with each viral proteins. So basically they identified 300 host proteins that bind to the SARS-CoV-2 proteins. And so among those proteins, sigma 1 and sigma 2 proteins were actually interacting. So they were the interactors. So of the virus life cycle. So basically these two proteins were interacting with the different stages of virus life cycles. Therefore, we can also target these sigma proteins to develop or to generate host-directed antiviral agents, guys. Okay. So now I have talked about, we can target different key molecules present in the host cells, which are really, really important for virus entry, replication, translation of the genome, virus assembly, and the release of the progeny virus particles. So we can target different important host molecules to have host-directed antiviral agents. Okay. So now, So now in this slide, I'm going to talk about what are the potential advantages. I also already mentioned few advantages already, and also disadvantages of host-directed antiviral agents. Okay. Okay. So one most important advantage of host-directed antiviral agents, as I mentioned already, is that they have broad spectrum of activity against different viruses that use the same host target. Okay, so if we have a host-directed antiviral agents against a host target, and that host target is same for different viruses, then we can use they have the broad spectrum of activity against different viruses that use the same host target. Okay, so this is one benefit. Another benefit is that, okay, for different genotypes and the serotypes, also we can use the same host-directed antiviral agents because they are also using the same host target. And also another important advantage is that they have a high barrier to the development of genetic resistance, because even if there is some mutation in the virus, that will not affect so much. Because we are targeting the host molecule, so the mutation in the virus will have minimal effect in the host-directed antiviral agents. So because therefore affording high barrier to the development of the genetic resistance, because we are targeting the host molecule, and if the host molecule, the virus will use the same host molecule, and if there is no mutation in the host molecule but only the mutation in the virus, then that also is not going to affect so much our antiviral agents, therefore providing high barrier to the development of genetic resistance. Another important advantage is numerous putative drug targets for cross-class combination therapy, and the most important thing is that if we have a new virus that is an emerging virus, or let's say potential epidemics or pandemics, but that uses the same host protein or the host target as the previous viruses, then we can already, these host-directed antiviral agents, they can be already available before even pandemics or epidemics. Okay, so that is also the major advantage of these host-directed antiviral agents. The next advantage is that the potential of preemptive development of agents before a realized viral threat. This I already discussed. Okay, so now what are then the potential risks associated with host-directed antiviral agents? One is potential of host pathway related toxicity. Okay, so this is one important risk, and another risk is that we are targeting the host molecule, and this host molecule may be important for the physiological functions of the host. So then that can also result in this molecule related toxicity. And another thing is that because the host or population specific polymorphism on different host target expression, because the target protein also have different polymorphism, host or population specific polymorphism, and in that condition then we cannot use the one host directed antiviral agents targeted against that particular target for the other population because of this polymorphism, host specific or the population specific polymorphism. Another important thing that I must mention is that the poor translation in vitro, okay, we might get better results, but when we test in vivo models, then we get poor translation of what we observed in vitro. So this is one risk. Another risk is that the complex mechanism of action, because deconvolution of target and target specific effects challenging. Okay, so in the host, the target molecule may be playing important role in different pathways and may be important for different physiological processes. Therefore, it might involve complex mechanism of action. Okay, so this point also we have to consider. Another important thing that we need to consider is the possibility of redundant host mechanisms that ease virus dependence on the selected target. Finally, what I would like to mention is that the host-directed antiviral agents, they may require directly acting antivirals as a combination therapy to achieve maximum benefits. Now, finally, to the proportion of virus versus host-directed antiviral agents. So as you can see in this graph that the directly acting antiviral agents, they account for approximately 89 to 90% of the antiviral drugs, whereas if we combine even these two as one class, interferons or host-directed antiviral agents, even though interferons have been represented here as a separate class, that only accounts for around 10%. So this chart is in reference to the antiviral agents targeting HCV, HIV, and other related viruses. So as you can see that the directly acting antiviral agents, they account for approximately 90% of currently available drugs, whereas host-directed antiviral agents, they account for approximately 10% of currently available antiviral drugs. I hope, guys, this video was helpful. Please like and share the video, subscribe our channel, and also please do support on PayPal, the link for which has been provided in the description. Thank you very much for your kind attention.